Hello and Assalamualaikum everyone. We are from Subgroup 7 and today we will present our journal reflection which is Microbial Bioremediation of Heavy Metals from Textile Industry Dye Effluents using Isolated Bacterial Strains.
The fellow group members involved for this presentation are Tominu, Verlen, Hasina, and myself, Ripka.
For this presentation, we will present our summary of research work, benefits, limitation, and suggestion for future improvement.
Presenting about the summary of the research work. Textile industries pose environmental and human health issues due to presence of heavy metals in the textile dye effluents discharged into the environments. Cadmium, lead, and zinc affect aquatic biological process by absorbing light in the water bodies. Accumulation of heavy metals in plants consumed by humans cause hepatic and renal system damage, mental retardation, as well as basal ganglia degradation in the brain. Even though there are conventional techniques to remove the heavy metals from the effluents, such as electrowinning and reverse osmosis, it requires high-skilled technicians, not to mention the process is highly expensive. Thus, in this study, applications of microbial bioremediation by using Escherichia coli, Salmonella typhi, Bacillus licheniformis, and Pseudomonas fluorescens on the removal of heavy metals from textile dye effluents were conducted. The objective to this study are to remove or decrease the content of heavy metals from the textile dye effluent samples through microbial bioremediation and determine the percentage degradation of the heavy metals by the microbial strains. The methodology includes sample collection at different areas of Komarapalayam town, situated in Namakkal district, Tamil Nadu, India. Then, physico-chemical characterization of the sample were taken such as chemical oxygen demand, biochemical oxygen demand, total dissolved solids, and heavy metals. Inoculum was then prepared using a loop full of bacterial cultures inoculate in saline solution before incubation process for 3 hours at 37 degrees Celsius. The degradation of heavy metals was observed after 5 days by assessing the content using atomic absorption spectroscopy. The tetrate dye was digested before filtered twice using Whatman No. 1 filter paper, and heavy metal assay using atomic absorption spectroscopy was performed. Through the analysis of physico-chemical characterization of the sample show high level of pollution using the standards of NUKS. There are pungent fishy odors and a lot of variation in the pH values indicating acidic and basic property. In addition, POD and COD values are also high. Biodegradation of cadmium content shows Bacillus licheniformis with maximum percentage of degradation, 98.34%, while Escherichia coli with the least percentage of degradation, 92.06%. Biodegradation of lead shows Pseudomonas fluorescens with maximum percent of degradation, 94.32%, while Escherichia coli is the least, 86.73%. Biodegradation of the zinc content shows both Bacillus licheniformis and Pseudomonas fluorescens with maximum percent of degradation, 96.14%, while Salmonella typhi is the least, 91.78%. In conclusion, through this study, it is proven that microbial bioremediation using Escherichia coli, Salmonella typhi, Bacillus licheniformis, and Pseudomonas fluorescens effectively decrease the heavy metal content with a maximum of 98.34% of cadmium, 94.32% of lead, and 96.14% of zinc. Thus, microbial bioremediation is advantageous to humankind as a cost-saving and efficient alternative in the treatment of industry's effluents.
There are some benefits of microbial bioremediation of heavy metals from textile industry dye effluents using isolated bacterial strains. First is, the microbial bioremediation is an effective approach for the elimination of heavy metals such as cadmium, lead, and zinc from textile industry dye effluents. Heavy metals need to be eliminated because the main mechanism of heavy metal toxicity include the generations of free radicals to cause oxidative stress, damage of biological molecules such as enzymes, proteins, lipids, and nucleic acids, damage of DNA, which is key to carcinogenesis, as well as neurotoxicity. Secondly, The microbial bioremediation is proved as an effective, eco-friendly, affordable technology for removal of heavy metals from the textile dye effluents. Thirdly, the microbes can tolerate against the heavy metals, and they are armed with various resistance and catabolic potentials. This catabolic potential of microbes is enormous and is advantageous to mankind for a cleaner and healthier environment through bioremediation. Besides, the microbes play a crucial role in the remediation of heavy metals and pollutants through biosorption. Biosorption is a process that utilizes biological materials as adsorbents, and this method has been studied by several researchers as an alternative technique to conventional methods for heavy metal removal from wastewater. Lastly, the microbial bioremediation facilitates in the treatment of textile dye effluents before release into the environment.
Although microbial bioremediation of heavy metals is environmental friendly and cost-effective, there are several limitations. Based on the journal, the microbes reduce maximum of 98% of cadmium, 94% of lead, and 96% of zinc from the effluent samples. In most of the cases, bioremediation technique can only decrease the level of heavy metals but not eliminate 100% of them. Moreover, bioremediation is sensitive to environmental factors such as temperature and pH value. This is because the microbes need optimum conditions to survive and carry out its microbial activities. Thus, manipulation of environmental parameters should be included to allow quicker microbial growth. Excessive heavy metal concentration is also one of the limitations of the technique. However, the effectiveness of bioremediation can be enhanced by addition of inorganic fertilizers, biosurfactants, bulking agents, and more. Another limitation of bioremediation of heavy metals is the non-biodegradability of heavy metals. In addition, there are possibilities where the products of biodegradation may be more toxic than its parental form, which lead to the production of toxic metabolites. Last but not least, the treatment period of bioremediation is more time-consuming than other remediation technologies.
For future improvement, researchers can conduct more studies on gene level to know bioabsorption potential. It's also important to find out more heavy metal resistant ability of microorganisms. Next, conducting more research to generate more technology that can treat textile dye effluents. After that, study for using chemical in treatment of textile dye effluent also need to be done. Lastly, further research on artificial textile wastewater can be performed to reduce environmental issues. And here are our references. Okay, that's all for our presentation. Thank you for your attention.